Hey there. Have you ever wondered how scientists predict the likelihood of inheriting certain traits like eye color or genetic disorders? Today we're diving into the world of genetic probability. We'll break down inheritance patterns, allele frequencies, and how to calculate the chances of passing on specific genes. Let's get started. Okay, first things first. What do we mean by the big words? No worries if you haven't encountered these before. Let's start with the term allele. You can think of alleles as different versions of a gene. Take eye color, for example. Let S represent the allele for brown eyes with B and the allele for blue eyes with B. Now, B and B are alleles of the same gene, but they're different variants. One important thing to remember is that you inherit one allele from your mother and one from your father, which means you have two alleles that make up your genotype. In our example, your genotype could be BB, meaning you have two brown eye alleles, or it could be BB, meaning you have two blue eye alleles. And finally, the combination of your alleles determines your observable traits, which we call the phenotype. So if brown eyes are dominant over blue eyes, both BB and BB genotypes will result in the same brown eye phenotype. Got it, good. Next up, we have probability. Basically, probability is all about figuring out the chance of something happening. It can range from 0%, which means it won't happen, to 100%, which means it definitely will happen. For instance, if you flip a coin, what's the chance of getting heads? It's 50%, since there are two equally likely possibilities, and only one of them is heads. So now that we know these basic terms, let's see how they work together. Say both of your parents had brown eyes. How likely is it that you'll also have brown eyes? Well, since we know that brown eyes are dominant, we can represent the eye color genotypes as follows. Your mother has two brown eye alleles, and your father also has two brown eye alleles. This means you have a 100% chance of getting at least one B, right? That's right. You inherited B from your mother and B from your father, making your genotype BB, resulting in brown eyes. But what happens when you have one parent with brown eyes and the other with blue eyes? Here's where the Punnett square comes into play. A Punnett square helps us visualize all the possible genotypes and their probabilities that offspring can inherit from their parents. Let's set this up. We'll list out the possible alleles that your mother can pass on down the top, and the possible alleles from your father down the side. So from the brown-eyed parent, you have the option of inheriting either a B or a B, and from the blue-eyed parent, you have the option of inheriting either a B or a B. Next, simply fill in each box by combining the allele from the top and side. This means you have a 25% chance of being BB, a 50% chance of being BB, and a 25% chance of being BB, which gives us a 75% chance of having brown eyes and a 25% chance of having blue eyes. And just like that, we've calculated the probability of inheriting certain traits. Okay, great. But what if we want to know about allele frequencies within a whole population? Talk about the Hardy-Weinberg principle. The Hardy-Weinberg equation is a tool used to determine the genetic makeup of a population based on allele frequencies. For those who aren't familiar with allele frequencies, don't worry. Essentially, it's a way of measuring how common a particular allele is within a gene pool. So to break it down further, we have two variables, P and Q. P represents the frequency of the dominant allele, while Q stands for the frequency of the recessive allele. The sum of both frequencies must always equal 1. So P plus Q equals 1. This is because every individual in a population must have two copies of the gene, either two dominant copies, two recessive copies, or one of each. To use the Hardy-Weinberg principle, you need some data. Let us say we're re-looking at a population of 1,000 individuals, and we notice that 16 of them have a homozygous recessive genotype. In this case, let's assume that the trait is controlled by a single gene with two alleles. If these 16 individuals are homozygous recessive, it means they have two copies of the recessive allele. Therefore, the frequency of the recessive allele, Q, is the square root of 0.16, which equals 0.4. And since P plus Q equals 1, the frequency of the dominant allele P is 1 to 0.4, or 0.6. So there you have it. Using the Hardy-Weinberg principle, we can easily determine the frequency of both alleles in the population. Now it's time to test your knowledge. Try this practice problem. 2 cystic fibrosis carriers have a child. What's the probability that the child has cystic fibrosis? Pause the video here if you want to try solving it yourself. Cut your answer. Okay, let's reveal the solution together. So the probability of their child having cystic fibrosis is 25%. You can figure this out by creating a Punnett square for 2 heterozygous parents. In this case, each parent has a 50% chance of passing on either A or A. By combining the alleles, we get the following genotypes: A, 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 and A. This means there's a 25% chance that their child will inherit the homozygous recessive genotype, which causes cystic fibrosis. Before we wrap up, let's discuss some potential pitfalls and tips to avoid confusion. When dealing with genetics, it's essential to keep in mind that not all traits follow Mendelian inheritance patterns. Sometimes things can get a bit more complicated. For instance, consider this image showing flowers with multiple colors. Can you guess how this is possible? If you guessed it's due to more than one gene, you'd be right. This is an example of a trait not following simple dominant-recessive relationships, showcasing the complexity of genetics. Another thing to watch out for is that genes located close together on the same chromosome tend to be inherited together, a phenomenon called linkage. This means they don't follow the expected Mendelian inheritance patterns and can throw off your probability calculations if you're not aware of it. Additionally, if your sample size is too small, it might not accurately reflect the expected genetic ratios predicted by probability. For example, if you flip a coin 5 times and get heads every time, does that mean the coin is biased? Probably not. But if you flip it 100 times and still only get heads 45 times, that's much closer to the expected 50%. And that's a wrap on calculating genetic probabilities. Do you feel confident in your ability to predict inheritance patterns? To keep practicing, check out the additional resources where you'll find more interactive problems and in-depth explanations. Thanks for tuning in, and happy learning. All right, future scientists, get ready to test your knowledge with an awesome dihybrid cross problem. 2 pea plants with the genotype YYRRR crossed. Y and R represent dominant traits while Y and R are recessive. So here's your question. What's the probability that their offspring will have both recessive traits? Pause the video here if you want to solve it before seeing the solution. Right. Go ahead and type your answer in the comments section below. We can't wait to see your responses. And don't forget to stay tuned for more exciting science challenges.